We have previously discussed the invention of MRI, and in particular that of anatomical brain imaging in the early 1970s. Now, over the past 15 years, further advances in the application and analysis of MR anatomical imaging, particularly that of the human brain, have taken place. One technique, invented in the early 1990s, now allows for the tracing of anatomical tracts within the brain and even imaging of the anatomy of peripheral nerves. This technique is called diffusion tensor imaging and is based on the use of the diffusion of water molecules to generate different pixel intensities. These different intensities can also contain information about the direction of water movement, and this information can be used to track large fibre bundles throughout the brain and in peripheral nerves. This remains the only technique available that allows us to track fibre bundles throughout the brain in living humans. In 1990, Professor Michael Moseley reported that water diffusion in white matter was anisotropic. That is, the water movement had direction. In essence, what Professor Moseley reported relied on the measurement of Brownian motion of water molecules. So when a water molecule is in cerebrospinal fluid, for example, it can flow with the same ease in any direction. In contrast, when a water molecule is between, for example, two axons in white matter, its movement along the length of the axon is easy, but its movement perpendicular to the axon is severely restricted. Using this water diffusion information, in 1992, Professor Filler and colleagues produced the first diffusion tensor image. This image was collected in a monkey, and in it you can clearly see on this coronal image a bright band on both sides of the brain. Now this is the location of the corticospinal tract. So clearly this technique can be used to locate large fibre bundles in living animals. This was an incredible step forward, as we can now begin to use this technique to track large fibre bundles in living humans with various clinical conditions. So we now have a technique that can be used to see large fibre bundles in the human brain. To use diffusion tensor imaging to explore fibre bundles, it is typical that a series of images in which water diffusion in 32 or more directions are calculated. These images are then compared to a reference image. Here is a typical series of images showing a T1 weighted anatomical, a baseline image, and multiple images in which water movement in various directions is calculated. Now the typical minimal voxel size for a diffusion image is about 2 by 2 by 2 millimeters using a standard 3 Tesla scanner. So the resolution of DTI images is less than that of a standard T1 weighted anatomical image. So using the 32 or more directional images, one can use a number of mathematical equations to calculate standard measurements. Now the two most widely used standard measurements are one of mean diffusivity and fractional anisotropy. So mean diffusivity is really a measure of the average molecular motion of water independent of any tissue directionality. It can be affected by the cellular size or integrity within a particular area. So anything that changes the physical structure of that area will alter the way in which water will move and therefore alter mean diffusivity. Of course, given the highly complex structure of the cerebral cortex and the tight packing of axons in white matter, this means that mean diffusivity is restricted in both white matter and in grey matter relative to cerebrospinal fluid. So similar to voxel-based morphometry analysis of grey matter images, Mean diffusivity can be used also in a relatively simple manner by comparing brain maps of mean diffusivity between groups, for example between a group of controls and a patient population. So once mean diffusivity maps are calculated, they can be spatially normalized to a standard template. Following this, is it possible to compare mean diffusivity of a particular voxel in controls and patients by using t-tests. This can create a map of the brain where mean diffusivity is different between two populations. If an area, for example, shows a difference in mean diffusivity in a patient group compared with controls, one could draw the conclusion that some type of physical difference exists in this particular region. For example, increased mean diffusivity may result from a loss of neurons, dendritic pruning, or inflammation, whereas decreased mean diffusivity may result from dendritic sprouting or even astrocyte activation. Whilst it is difficult to determine the exact cellular changes responsible for a change in mean diffusivity, combining mean diffusivity results with other MRI markers such as grey matter volume or activity, and with results from experimental animal studies in which cellular changes can be explored, can result in a more complete picture of the changes responsible for the initial change in mean diffusivity. Now, in addition to mean diffusivity, fractional anisotropy is another measure commonly calculated from diffusion tensor images. Fractional anisotropy is a scalar value between 0 and 1 that is a measure of anisotropy. A value of 0 means that diffusion is isotropic, that is, it is unrestricted in all directions, whereas a value of 1 means that diffusion occurs only along one axis and is fully restricted along all other directions. So in most white matter, axons are aligned parallel to each other, and water molecules between these axons can diffuse along the length of the axon with relative ease, but they cannot diffuse perpendicular to the axons. This results in high fractional anisotropy. In contrast, in grey matter, water does not move in any particular direction with such ease, and so grey matter typically has low fractional anisotropy. So in grey matter, water can diffuse in all directions with similar ease. That is, it has isotropic diffusion. In contrast, in white matter, water can diffuse primarily along the longitudinal axis of the axons. It therefore has anisotropic diffusion or high fractional anisotropy. So like mean diffusivity, fractional anisotropy can be used in a relatively simple manner by comparing brain maps of fractional anisotropy between groups. The interpretation of fractional anisotropy can be a little bit more complicated, as it can mean a change in the direction of water movement and or the ease of this movement. For example, in white matter, a decrease in fractional anisotropy could result from myelin degeneration, so that water can move more freely perpendicular to the axons. One of the more exciting analysis procedures that can be performed using diffusion tensor images is that of tractography. As mentioned previously, water diffusion can be isotropic, where it flows in any direction with ease, or it can be anisotropic, where it flows primarily in a particular direction. Highly anisotropic flow or high fractional anisotropy results from water that can flow almost exclusively in a particular direction, as occurs between axons within a large fibre bundle. Now, the direction in which water diffuses more freely is called the principal eigenvector, and these eigenvectors can be color-coded to reveal a map of the brain in which the color of each voxel indicates the strength and direction of water movement. So in this axial slice through the brain, the green areas are regions in which water is diffusing almost exclusively in an anterior-posterior direction. In contrast, the red areas indicates areas in which water is diffusing most easily in a medial to lateral direction, Using the assumption that the principal eigenvector is associated with the direction of fiber bundles, one can then start to track large fiber bundles throughout the brain. This technique works remarkably well, and it is possible to track most of the fiber bundles in the living brain of humans. Here is an example of how tractography can be used to track a large fiber bundle in the human brain. By instructing the program to display fibers that pass through the posterior limb of internal capsule, the cerebral peduncles, and the pyramids, we can track the corticospinal tract from its origins in the primary motor cortex, through the corona radiata, the internal capsule, the cerebral peduncle, and through the pons. One could then use the same procedure to track the corticospinal tract, for example, in a group of patients following spinal cord injury. As has been shown in this investigation, the corticospinal tract displays lower fractional anisotropy and lower mean diffusivity in individuals after spinal cord injury. Now, these measurements could be used to track disease progression or recovery following a traumatic spinal cord injury. Here's another example of how tractography can be used to explore brain anatomy in living humans. In this study, the authors tracked the arcuate fasciculus in children. Now, this is a large fibre bundle that connects two important areas for language, Broca's area and Wernicke's area. And it was found in this study that fractional anisotropy levels within this bundle were associated with the children's reading skills. Now, while these are just two examples of how tractography can be used to explore brain structure, many tractography studies have been performed over the past 5 to 10 years. These studies have tracked many fibre bundles, including those involved in motor and sensory function, higher order emotional processing, memory, and even tracks in the brainstem, spinal cord, and in peripheral nerves. Combining this technique with other brain imaging modalities, such as those that measure regional brain activity and biochemistry, can provide a more complete picture of brain anatomy and function in both healthy individuals as well as in those individuals with various conditions.